are proteins that are made at the ribosomes that are considered catalysts. And catalysts mean that they speed up chemical reactions, they initiate or get the reactions going, and they're what I call the cell playmakers. All chemical reactions that take place in cells most likely have an enzyme that's involved. So they initiate or start chemical reactions within cells. They are considered catalysts. And if you're a catalyst, maybe the reaction would have already occurred, but you're getting it to go along a lot faster. So you're speeding up that chemical reaction. So a good example is digesting your food. If you didn't have enzymes, then your teeth would mechanically break down the food. Maybe it's a piece of steak or a hamburger and you're chewing it, but you would only rely on the physical mashing and chewing to be able to break it down into smaller pieces, and maybe some acid in the stomach. But enzymes are specific to what they can attach to and break down. So if it's a specific enzyme that breaks down protein, then it can only break down protein. It can't break down other things that you might eat. So digestion would take place, but it just would be a lot slower. So they are considered catalysts, which means they speed up chemical reactions, the rate at which it can happen. So that's to your advantage if you want to do more work. They make the reaction take place at a faster rate to make them more efficient. And also, they lower the amount of energy needed for the reaction to happen. So I would compare it to a gallon of gas that you put in a Hummer that might get 16 miles to the gallon at best. And then you put the same gallon of gas in a Prius that may get over 50 miles to the gallon. So you still have the same amount of energy, but the Prius can actually transform it and do more work than the Hummer can. It takes it farther. It can burn and burn that gasoline at a more efficient rate. So enzymes help these chemical reactions in your body in your cells to be able to go and take place more efficiently with less energy. So that's the next thing that we'll add, is that they lower the energy needed for the reaction to take place. So they lower the energy needed for the reaction to occur. And that is decrease or lower the activation energy. So while they speed up reactions, they lower the energy needed. So it doesn't take as much. All right, so the next thing is enzymes are specific shapes. So just like the key that fits into the lock of your door of a car or the door to your home, that key can only fit that lock and open that lock. So it has a specific shape. Now, sometimes keys you can kind of force it into a lock and it may go, but there's a special little sweet spot where it can really lock in. And that's when you know that it's going to open the door, the doorknob, or that it's not going to get stuck. So you know that it's actually going to open the doorknob. So we're going to draw what an enzyme could look like and then what the substrate could look like, which is what it locks into. So it's like two pieces of the puzzle coming together and they fit just perfectly. So they have a specific shape that attaches to a specific, and it's called substrate. That's what it attaches to. Okay. So what I'm going to do is now draw a picture below. I'll put it here so you can see. So let's say that the enzyme is shaped like this, like a Pac-Man. So this is an enzymatic reaction. This is just like a generic reaction. Okay, so here's the enzyme. And it's going to fit into and bind to a substrate. And they're going to fit together at that little sweet spot, that little active site that is the perfect little place where those two merge together or combine. And that's called the complex, the enzyme substrate complex. But what I'm going to do is color this little area where the two fit together, and that's called the active site. Now, once they fit together just perfectly, the enzyme will change shape and it'll twist it, and it can actually break the substrate apart. So the enzyme is recycled. It's left to do more work and to attach to more of those same substrates. But now the substrate has been broken into products. So here's a real life example. So there's an enzyme in your spit called amylase. We'll put in spit. Okay, so here's the enzyme. And I don't know what it looks like, but we'll say that it looks like this. Here's the enzyme amylase. It attaches to starch. Once it attaches to that starch at the active site, then it is able to digest, if given enough time, it's able to digest that starch into glucose, which you know is a smaller sugar, and fructose, which is also a small sugar, a one-ring sugar. So it's able to digest it into smaller pieces. The advantage of that is that these little pieces can now move through the cell membrane and be used by the cell. For glucose and fructose, that's used by the mitochondria to make ATP. So this is why we have a digestive system that has all these different enzymes that it can attach to different parts of what we eat, what we consume, and be able to break it into different usable pieces. So that's how an enzyme can work. So the next thing is, as far as enzymes go, is sometimes things interfere with their ability to work. So enzymes, so what affects an enzyme's ability to do its job? So ability to function. One thing is if you is the amount of substrate you have. If you have a lot of substrate, then it can break a whole lot down. It can do more work. So so it's called substrate concentration. So if it has a lot, this is the enzyme. If it has a whole lot of substrate to break down, then it's going to have a higher rate. It can do more work. Another is high temperatures. If we heat up that enzyme, it can actually cause the enzyme to change shape and that it can no longer fit into its substrate. So if the enzyme is exposed to high temperatures, then it would be like it's shape changing, and now this will not fit into this. And that's called denaturing. So at high temperature can actually cause the enzyme to change shape and the reaction rate will stop. So high temperatures can change the shape of an enzyme. That it will no longer fit into the substrate. So it can't do its job. And that's called, it's denatured. So it's denatured. So this would be, if it looked like a Pac-Man, now it might look like a blob. Okay? So that can't fit into its substrate. So that's the second way. A third way is cold temperatures slow down the reactions. And that's because the molecules are moving slower, so the enzyme moves slower to get to the substrate, and it moves slower to pull it apart. So it can, while it can still bond to the substrate, it just takes it longer to do that. So it's, if you're really cold, you may not have as much energy to actually move around, and that's true with an enzyme too. So cold temperatures can slow down the reactions because the molecules move slower. Okay, and then a fourth way is that we can denature the enzyme and change its shape again, is to add a very strong acid. And denature or change the shape of an enzyme. Okay. So that is the skinny on enzymes.